Okay, let's unpack this. Imagine trying to build a fully formed, complex creature, a bird. No less starting from just a tiny speck of cells.
Right.
And not just any development, but one happening inside an egg, which is almost entirely dominated by a massive inert yolk.
Mm-hmm.
Sounds like a biological engineering nightmare, right?
Mm-hmm.
Well, today, we're taking a deep dive into how the bird embryo pulls off this incredible feat. The early development of life within that shell.
It really is something.
We're going to explore the sheer ingenuity of how a seemingly simple, flat sheet of cells on top of that yolk manages to organize itself, lay down its basic body plan, and then remarkably builds its own complete life support system, all within the confines of that egg.
Yeah, it's a masterclass in developmental biology, really showing how life adapts to truly unique constraints.
Absolutely.
What's truly striking here is understanding the specialized adaptations birds have evolved for gastrulation and early structural formation, especially when we compare them to other animals. We'll literally trace the journey from those initial layers of cells to a fully three-dimensional embryo, highlighting how essential structure and function are established, ready to thrive within its shell. You've given us some excellent source material, detailed lecture notes and diagrams, which will be our essential blueprint today.
Great. So let's jump right into the first big challenge, gastrulation. We've talked about this process before, how cells rearrange to form the fundamental germ layers.
The 3 layers.
In simpler organisms, like a sea urchin, cells might in-pocket or invaginate directly. Even amphibians, with their larger yolky cells, have a distinct process where cells migrate through a narrow slit.
Different strategies, yeah.
But a bird egg is practically a giant sphere of nutrient-rich yolk. I'm struggling to picture how gastrulation even begins when the main event is just this enormous yellow mass.
That's precisely the right question to ask because the sheer quantity of yolk is the ultimate constraint. It completely prevents the typical invagination we see in simpler embryos.
Right, it just gets in the way.
Totally. Instead, the bird has evolved a remarkably different, and I'd argue, incredibly ingenious strategy. Think of it this way. Instead of a full cellular sphere, cleavage, the initial cell division, occurs only in a sort of little cytoplasmic cap, right on top of that massive yolk.
Just on the surface.
Exactly, just a tiny disk of activity. This tiny cap, known as the blastodisc, then cleaves to form a multi-layered structure called the blastoderm. Now, the blastoderm isn't directly on the yolk. It sits above a subgerminal space that provides a crucial separation.
Okay, a little buffer zone.
Precisely. And the first real step of gastrulation in a bird embryo is when this blastoderm separates into 2 distinct layers, an upper epiblast layer and a lower hypoblast layer. The critical space that forms between them is the cleft space. This isn't just an empty void, it's the staging ground, the waiting room, where incoming cells will soon arrive to differentiate.
So if I'm picturing this correctly, we start with a cellular pancake on top of the yolk.
Kinda, yeah.
And then it splits into an upper and lower layer with a crucial new space in between. Okay, that makes sense for starting the layering process. But how do these flat layers then truly get organized into the 3 fundamental germ layers, ectoderm, mesoderm, and endoderm, that will build the entire chick?
Right, that's the next puzzle. This is where the bird embryo performs what can only be described as a biological trick. The second critical step involves lateral cells moving inward towards the center of this blastodisc. This movement results in the formation of a thickened structure known as the primitive streak.
The primitive streak, okay.
This streak is functionally central to bird gastrulation. It's the main highway for cell migration. Imagine a distinct indentation, almost like a seam, appearing right down the midline of this primitive streak that's the primitive groove.
Like a little trench forming.
Exactly. And this primitive groove functions similarly to the blastopore of other embryos, but instead of being a simple opening, it acts as the primary site of cell ingression.
So cells dive in there.
Precisely. As the primitive streak elongates, cells from the epiblast surface literally dive through this primitive groove, moving into that cleft space we talked about earlier. Some of these incoming epiblast cells will form the endoderm by displacing and integrating with the hypoblast, while others form the middle mesoderm layer.
And the ones left on top.
The remaining epiblast cells become the ectoderm, so you get all 3 layers established from that migration.
Wow.
What's truly ingenious here is that in the bird, gastrulation occurs only in this non-yolky cell cap. The yolk itself remains entirely uncleaved and uninvolved in the actual cell rearrangement.
It just sits there providing fuel.
Basically, yes.
Yeah.
If we step back and look at the broader evolutionary canvas, this isn't just an adaptation, it's a masterclass in biological resourcefulness. The bird embryo essentially created a mobile command center for development atop a colossal fuel tank.
Huh. That's a great way to put it.
Proving that developmental complexity isn't limited by sheer volume, but by ingenious spatial organization and metabolic efficiency.
That's quite the workaround. So the bird ingeniously bypassed the yolk problem by doing all the heavy lifting in that tiny cell cap. Now we have our 3 fundamental germ layers, ectoderm, mesoderm, and endoderm, all laid out thanks to that amazing primitive streak.
Right. The foundation is set.
But it's still essentially a flat blueprint, a sheet of cells. It feels like we've solved one puzzle only to face another. How does this flat sheet actually transform into a 3-dimensional chick, a complex, rounded organism that will eventually fill out its shell? Does it just, like, inflate?
No, it definitely doesn't just inflate. This is where the embryo engages in a series of impressive body folds to literally lift itself off the yolk and take on a 3-dimensional shape.
Body folds. Go.
It's not just growing, it's actively sculpting itself.
Yeah.
These folds address the fundamental challenge of how a flat gastrula achieves complex morphology. Think of it as an intricate biological origami.
Origami, I like that.
The embryo transforms from that layered sheet of cells atop the yolk into a cylinder by undergoing a series of folds that occur within the embryo proper.
So the embryo itself is folding?
Yes, the main part of the embryo. We see a distinct head fold forming at the anterior or front end of the embryo.
Okay.
At the opposite posterior end, a tail fold emerges. And along the sides, you have lateral body folds.
Head, tail, and sides folding. Upwards.
Downwards.
Kind of folding under, lifting the main body up off the yolk. As these folds deepen and progress, the embryo profoundly changes its form, becoming a 3-dimensional cylinder, now distinct and elevated above the yolk.
So it goes from flat pancake to like a tube.
A tube or cylinder, exactly. It's worth noting that even at this stage, the central mid-gut region of the embryo remains open to the yolk, maintaining its crucial nutrient connection.
So it's still connected for food.
Absolutely. And of course, these body folds work in concert with neural folds, which are simultaneously forming and are absolutely integral to the development of the nervous system. That's a whole other fascinating process happening at the same time.
Right, building the brain and spinal cord. Exactly. What's particularly striking is that very similar foldings occur in both reptilian and mammalian embryos.
Really? Even mammals?
Yes. This illustrates a profound common evolutionary strategy for establishing the basic vertebrate body plan, even when comparing vastly different reproductive modes like egg-laying versus live birth. These folds are more than mere shaping. They're an ancient, elegant blueprint for building a complex 3D organism from a simple sheet. They illustrate a fundamental principle, how mechanical forces and precise cellular movements orchestrate life's most intricate architecture.
That's fascinating, an ancient universal sculpting technique. So our little cylinder is now taking shape, looking more and more like a tiny developing organism.
Getting there.
But it's still sealed inside an eggshell. It needs constant protection, and it needs a sophisticated way to manage waste and breathe for weeks. What does this all mean for its survival within that confined, self-contained space? It can't just sprout lungs and a kidney overnight.
You've hit on the next critical challenge. And you're right, it needs a whole support system. Once the embryo proper starts to form, another crucial series of folds kicks in. But these are in the cells surrounding the embryo, not the embryo itself.
Outside the main body.
Exactly. These are the extra embryonic folds, and they are responsible for creating specialized membranes that become the chick's essential life support system, its protection, its waste management, and its respiratory system.
The life support system. Okay.
Yeah.
How does that work?
Let's break it down. We have an anterior extraembryonic somatopleur that's a specific combination of ectoderm and mesoderm, which starts to fold over the head of the embryo.
Folding over it.
Yes, like a hood. Similarly, a posterior extraembryonic somatopleur folds over the tail end, and complementary lateral folds also occur along the sides. These specific folds are the developing folds of the amnion. They extend to completely cover the embryo from all directions and eventually fuse together over the top.
They meet in the middle.
They meet and fuse, forming what looks like a double helmet that encases the embryo.
A double helmet.
Yeah, it's layered. This helmet is incredibly clever. The outer layer is called the chorion, and the inner layer is the amnion. Both are composed of that same somatopleur material.
Okay, chorion outside, amnion inside. Got it.
And this sophisticated wrapping creates two absolutely vital spaces. The extraembryonic chorion is the space nestled between the amnion and the chorion.
Between the two helmet layers.
Right. But even more critical for the embryo itself is the amniotic cavity, the space directly between the amnion and the embryo proper.
Space right around the embryo.
Exactly. This naturally leads us to ask, why is the amniotic cavity so vital? It contains amniotic fluid, which plays two crucial roles. It protects the embryo from drying out in its enclosed environment, and it provides a cushion against mechanical injury.
Ah, so it's like a shock absorber.
It's the ultimate internal shock absorber and humidifier, providing a stable, protective environment for weeks on end.
So the amnion is like its personal protective, climate-controlled bubble, keeping it safe and hydrated. That's incredible foresight from nature.
It really is.
But what about the less glamorous, but equally vital tasks of getting rid of metabolic waste and, crucially, breathing inside a sealed shell with no direct connection to the outside world? I can't just hold its breath for 3 weeks.
Exactly. Waste disposal and breathing, key problems. For these vital functions, we introduce another important sac, the allantois.
The allantois.
This develops as an outpocketing of the hindgut, so from the back end of the digestive tract, and it consists of splenchnopleur, which is a combination of endoderm and splenchnic mesoderm.
Another membrane combo.
Right. The allantois extends outward into the extraembryonic column, that space between the amnion and chorion, growing larger and larger, and it serves as the embryo's primary excretory organ, where uric acid, the bird's waste product, is safely deposited and stored away from the developing chick.
So it's basically the waste storage tank.
Essentially, yes. Keeps the main environment clean. But here's where it gets truly fascinating. This allantois, as it grows, fuses with the chorion, that outer layer of the helmet we discussed.
The outermost layer.
Fuses with it to form what's called the chorioallantoic membrane.
Chorioallantoic, okay.
What's particularly ingenious is how this combined membrane becomes densely filled with blood vessels and functions as the primary respiratory organ for the entire period until the chick hatches.
The breathing organ. How?
Well, oxygen from the outside passes through the porous eggshell, diffuses across the shell membranes and into these chorioallantoic blood vessels, and is delivered to the chick.
Through the shell. Wow.
Yep. Conversely, carbon dioxide, the waste gas from the chick's metabolism, passes out of the vessels, across the membranes, and through the shell to the outside.
So it literally allows the chick to breathe through its shell.
Precisely. It creates a completely self-contained yet highly efficient gas exchange system. It's an engineering marvel built cell by cell.
Wow, what an incredible journey we've taken today. From a minuscule cap of cells on a giant yolk.
Seems like ages ago now.
Through the intricate dance of gastrulation and the amazing body folds that give it shape, to the ingenious construction of a sophisticated, multi-layered life support system with the amnion, chorion, and allantois.
Yeah, the whole package.
We've truly seen how the bird embryo handles every challenge thrown its way by its unique environment within the egg.
If we connect this to the bigger picture, this deep dive vividly demonstrates that while different organisms employ diverse patterns for gastrulation and early development, be it invagination, cell migration through slits, or the primitive streak, the fundamental outcome is always the same.
Building the body plan.
Exactly. Rearranging cells with incredible precision to give them form and structure, establishing the basic body plan. The bird embryo's adaptations are a testament to the elegant and diverse solutions evolution finds to allow life to flourish under unique and often profoundly challenging circumstances like developing inside a yolky egg.
Yeah.
It underscores biology's ultimate problem-solving capacity.
So what does this all mean for you listening? It's a powerful reminder, I think, of the sheer ingenuity and adaptability of life.
Definitely.
Every creature, from the simplest to the most complex, has a developmental story that's nothing short of miraculous, a testament to complex biological programming making the most of its environment. It shows us that constraints often breed the most creative solutions.
That's a great takeaway. And this deep dive into the bird embryo really makes you wonder, doesn't it? Given the remarkable complexity and self-sufficiency of development within an egg, building its own entire world, what might have been the evolutionary pressures or advantages that led to the development of live birth viviparity in other vertebrate lineages, like mammals?
Good question.
What critical trade-offs were made by moving away from this elaborate self-contained egg system? And what new challenges or opportunities did that present for how new life comes into the world.
Yeah, what did giving up the shell gain you, and what did it cost? That's something to chew on. Thank you for joining us on this deep dive into the remarkable world of bird embryology. Keep exploring, keep questioning.
Always keep questioning.
And we'll see you on the next deep dive.